Fertilization is a process in which the sperm and the egg unite in the fallopian tube. After the meeting of the sperm cell and the egg cell, the zygote will be formed and will be divided into two. But why does a zygote needs to be divided, and how does it happen? Humans have 46 chromosomes, and that's 23 chromosomes from male and 23 chromosomes from female, while cats have 38 chromosomes. Dogs have 78 chromosomes, and horses have 62 chromosomes. After the combination of paternal and maternal chromosomes, the embryonic genome will also be activated, which will happen at species-specific stage. Embryonic genome activation happens in two phases, the minor and the major genome activation. In mouse, the minor genome activation happens at late first cell cycle, while the major embryonic genome activation happens at the second cell cycle. The zygote will undergo several mitotic divisions and start to replicate the DNA. The process is called cleavage. Cleavage is a process of repeated mitotic divisions that will result in the increasing number of cells. The cleavage patterns of an individual species can be determined by the amount of yolk in the fertilized ovum. Myelicital eggs contain a small amount of evenly distributed yolk. These eggs are found in eutherian mammals. Mesolicital eggs, on the other hand, have a moderate amount of yolk distribution. Many amphibians' eggs are of this type. Lastly, when there is a large amount of yolk present, this is referred to as a megalithical egg. This category includes reptiles. Cleavage can be classified in several ways based on the abundance and distribution of the yolk. The term holoblastic cleavage refers to the process by which the entire ovum is completely divided, whereas the blastomeres can be either equal or unequal. While blastomeres in myelicital eggs are approximately all the same size, placental mammals are an example of this type. In medialicital eggs, blastomeres of an unequal size are produced. Plastic cleavage occurs in megalicital eggs because mitosis is restricted to the animal pole, where the cytoplasm is devoid of yolk. The vegetal pole's inner yolk mass does not divide. Blastomeres are the two daughter cells formed during the cleavage process. Following the division, the two daughter cells will begin to divide again, following the same procedure as how the zygote divided into two, resulting in a 4-cell embryo. The 4-cell embryo will be divided again into an 8-cell embryo. The cleavage process will continue until the morula is formed, which is a mulberry-like structure that is Latin for mulberry. But even though the cell divides and they increases, there is no growth. The cell continues to divide and they get smaller and smaller, but they are still equal in size and have the exact genetic information. So back to the question, what is the purpose of the division of a zygote? It is to divide the zygote into many identical cells that could later be used as the building blocks of an adult organisms.
Outer cells constitute the tropectoderm or trophoblast. Tropectoderm will be used before plasmentation and trophoblast will be used when this cell became engaged in placental formation. The form attached between neighboring cells of the tropectoderm result from specialized intercellular junction including light junction and demosomes. There are some species differences in the timing of compaction. In the pig, it occurs every earlier in development, around the 8th cell stage, whereas in a cattle it happened later, around the 16 to 32 cell stage. At the molecular level, differentiation of other blastomeres, at least in the mouse, appear to rely on downregulation of the transcription factor October 14 followed by an upregulation of other transcription factors such as CDX2 and eos mesodermin.
Compaction of the morula is a prerequisite for subsequent blastulation, the formation of a central fluid-filled cavity, which is the blastocyst cavity within the early embryo. Meaning to say, blastulation will not occur without the compaction of the morula. So what does it mean when we talk about blastulation? Blastulation transforms the embryo into a blastocyst, usually within the uterine lumen during the first week of development. So in this figure, you can see the pig blastocyst at day 6 of development as seen through the stereo microscope. Blastulation is brought about mainly by the tropectoderm and its control of fluid transport into the blastocyst cavity. Eventually, the inner blastomers become positioned at one pole of the embryo, forming the inner cell mass or the ICM, as you can see in this figure. Descendants of the ICM will form the embryo proper, while the cells of the tropectoderm will give rise to the embryonic part of the placenta. So we will learn more about placenta in chapter 9. We must take note that the ratio of cells in the ICM to those in the tropectoderm is around 1:3. Also, the portion of the tropectoderm covering the ICM is referred to as the polar tropectoderm, whereas the rest is known as the moral tropectoderm. So as we can see here in this figure, this is the moral tropectoderm and this is the polar tropectoderm. As we proceed, did you know that with the osmotic pressure inside the blastocyst cavity rising, the blastocyst gradually expands? Upon flushing of a blastocyst from the uterus, for embryo transfer for example, it is common to see the blastocyst collapse and expand several times. Whether this is an in vitro artifact or a normal physiological phenomena also occurring in the uterus is unknown. Eventually, expansion of the blastocyst leads to rupture of the covering zona pellucida, which allows the blastocyst to escape through the opening. You can see in this figure the hatching of pig blastocysts, and here is the zona pellucida. In some species, this process, which is known as hatching, is aided by proteolytic enzymes released from the endometrium acting upon the glycoprotein forming the zona pellucida. So what is proteolytic enzymes? It is also known as proteases, and these are enzymes that break down protein. So we tackle these enzymes way back in biochemistry. So in this case, the proteolytic enzymes will break down the glycoprotein forming the zona pellucida. So if the zona pellucida rupture, this will allow the blastocyst to escape through the opening as I mentioned earlier. In the horse, a compensatory capsule forms between the tropectoderm and the overlying zona pellucida before the zona is shed. This means, even before hatching, the blastocyst becomes surrounded by a tough elastic glycoprotein capsule produced in a large part by the tropectoderm inside the zona pellucida. So we can see the capsule here. So next, in contrast to other species, the conceptus, due to the presence of the capsule, remains spherical during restoration until around day 21 when the capsule is lost. So we can see in this figure an early illustrations of the queen capsule. Here is a 14-day queen conceptus illustrated by Crowling in 1937, and it clearly showing the capsule labeled as CP or zona pellucida. Here. We must also take note that the capsule plays an important role in the maintenance of early pregnancy. Why? It is because the capsule allows the spherical embryo to traverse the entire uterine lumen during day 6 to 17, providing the signals essential for pregnancy maintenance. So this will be discussed more in chapter 9. Around the time of hatching, the ICM differentiates into two cell populations. Those facing the blastocyst cavity become flattened and delaminate, forming an inner cell sheet referred to as the hypoblast, whereas the remaining cells form the multi-layered epiblast. Let's remember, this is the hypoblast and this is the epiblast. Furthermore, small intercellular cavitation may arise here in the epiblast. The hypoblast, like the tropectoderm, is epithelial in character, Gradually, it forms a complete inner lining beneath not only the epiblast but also the tropectoderm. In the horse, the hypoblast first forms separate colonies before coalescing into an internal closed compartment. We can see a border here. This is the hypoblast. In either case, a cavity referred to as the primitive yolk sac is formed. So, this is the primitive yolk sac. Next, let's take this figure as illustration. At the molecular level, the expression of the transcription factor NANOG is essential for the cells forming the epiblast, at least in mice, while the transcription factor GATA6 is a key regulator of hypoblast formation. The epiblast will later form the embryo proper, whereas the hypoblast will form the inner epithelium of the yolk sac, which, depending on the species, may become engaged in placentation. So we will also tackle this more in Chapter 9. However, at least in the mouse and rabbit, the hypoblast has also been shown to play a significant role in regulating the survival, proliferation, and differentiation of the overlying epiblast. This regulation is in part mediated through formation of a basement membrane between the hypoblast and the epiblast. We can see here there's a line between the epiblast and hypoblast. This is the basement membrane. As we proceed, in the domestic species, the polar tropectoderm covering the epiblast, which is known as Robert's layer, gradually disintegrates. So we can see in this figure that Robert's layer is in the process of degenerating. So when it is completely degenerate and it's lost, it leads to exposing the epiblast to the uterine environment. So here is the epiblast, and this will be exposed to the uterine environment here. Before the shedding of Robert's layer, tight junctions are formed between the outermost epiblast cells to seal the embryo despite the loss of the polar tropectoderm. We must take note that after the loss of Robert's layer, the epiblast is clearly discernible as a loosened structure, circular at first but becoming oval. Together with its underlying hypoblast, this structure is known as the embryonic disc. The embryonic disc is in the box labeled with B and is seen at a higher magnification in B here. During the formation of the embryonic disc, the blastocyst is still expanding. Once the disc is formed, the tropectoderm with its underlying hypoblast becomes reshaped and the embryo becomes ovoid. So if we say that it is ovoid, meaning it is like egg-shaped, just like in this figure. Next, in the ruminants and pig, the process of elongation continues and the embryo becomes first tubular and later filamentous. So what does it mean when first tubular and later filamentous? As you can see in this figure, this is the tubular embryo at day 11 of development. The tubular embryo is bit folded. On the other hand, here is the intermiggled filamentous embryos at day 13 of development. So if we say filamentous, it is thin in diameter resembling a thread. So the total mass of the embryo does not increase at the same rate as its length, and so the embryo becomes thread-like and tremendously long. This phenomena is particularly pronounced in the pig, where the embryo develops from a sphere about 1 cm in diameter on day 10 of development to a filamentous structure about 1 meter long on day 13. Elongation is particularly dramatic on day 12 and 13, wherein it ranges from 30 to 45 mm per hour. This increase cannot be explained by mitosis alone, as it involves the structuring of self-cytoskeletons and shape as well. Lastly, in ruminants, embryo elongation is less pronounced. In cattle, the length increases by up to 35 cm between days 12 and 21. So here is the elongation of bovine embryos around day 16, day 18, day 22, and day 27 of development. We notice that from here, the the embryo elongates as days goes by. Take note, in contrast to the ruminants and the pig, in the horse, the embryo does not elongate but stays spherical during this period of development.